Hey guys, welcome back to my channel. So I hope you have seen my previous video where I have shared some previous year questions of microbiology which have been asked in GATE Life Sciences exam previously, and in this video I would be sharing some more questions for the same exam. Okay, so if you haven't subscribed to my channel yet, then please do subscribe to my channel and do share my video with your friends who are preparing for this particular exam. Okay, so let's start with this video. First question says folic acid synthesis in bacteria is competitively inhibited by sulfonamides. Which of the following essential components is replaced by sulfonamides during the process of folic acid synthesis? So here what we are talking about is folic acid synthesis. So let's just decode this question, as this question is a bigger one. So here it is saying, the question is saying folic acid synthesis which is taking place in bacteria, it is being competitively inhibited by sulfonamides. Now we have to tell the essential component which is replacing sulfonamide during the process of folic acid synthesis. So we have the options pteridine, glutamic acid, para-amino benzoic acid, or sulfa-methoxazole. Okay, so we have to tell the correct option or the component which is replacing sulfonamides. So para-amino benzoic acid is replacing sulfonamides during the process of folic acid synthesis. So the option C is the correct answer. Now other question is, which of the following cellular structures are absent in eukaryotes and are present at least in some prokaryotic organisms? So we have to tell the cellular structures which are absent in eukaryotes but are found in some of the prokaryotic organisms. So we have respiratory enzymes, ribosomes, nucleoid, internal membranes, or pili. So we have nucleoid and pili that are present only in the prokaryotic organisms. So the option goes, the option is option third and fifth. Option A is the correct answer over here. Okay, so nucleoid and pili are present only in the prokaryotic organisms. Now we have a match the following in which we are having antibodies and we have the description or function. So IgA is responsible for the mucosal immunity. Okay, the immunity that is provided by the mucosal lining of our internal organs. Now IgG is responsible for the most abundant the most abundant antibody in the blood. IgD is found in the B cell membranes or found on the B cell membranes. IgE is the immunity to the parasitic infections or the allergies. Okay, so the option C is the correct match the following answer. Now we have granules are inclusion bodies that are found in bacteria. Which of the following statements are true about granules? First statement says granules can be storage vessels for carbohydrates and lipids. Some granules supply substances for a variety of metabolic processes. Granules are dissolved in cytoplasm and granules are membrane enclosed structures. So we have to tell which of the following given statements are true. So the statements granules can be storage vessels for carbohydrates, lipids. So this is true. Then some granules supply substances for a variety of metabolic processes. This one is true. So you can see that option C is having first and second statements. So option C is the correct answer over here. Now among the following characteristics listed below, select those that are the most appropriate for the archaebacteria. Okay, I hope you have studied about the archaebacteria. So the statements that are or the characteristics that are given are peptidoglycan containing muramic acid and D-amino acids are present in the cell wall. This is not the most appropriate characteristic of the archaebacteria. Then long chain branched alcohols which are phytanols bound to glycerol by ether linkages are found in the lipids of cytoplasmic membrane. This is the characteristic that is being found in archaebacteria. First amino acid to initiate new polypeptide chain is N Formylmethionine. This is not the one, this is not the characteristic. Okay, then we have the fourth one, which is translation process, is sensitive to the action of diphtheria toxin. So second and fourth are the characteristics that are present or found in the archaebacteria. So for attempting this question correctly, you must have read about archaebacteria in detail. Do stick to what is being asked in your syllabus. Do look at the syllabus after looking at this question and see what you have in regard to the archaebacteria in your syllabus, and see how much this question matches to what is being given or being asked for you to study in the syllabus. Okay, then you will find that some of the topics need in-depth study, like the types of bacteria, what are their characteristics, what are their different metabolic processes that are occurring inside them, what are their different internal structures or the external structures. So you must be familiar about them. If you are familiar or at least 80% of the information that is being asked in your syllabus, if you know that, okay, you would be freely attempting the questions correctly. Okay, so for the confidence to attempt the questions, first go through the previous year questions. So I'll make sure I could share with you as much questions as I can. So you would be having a good stock of previous year questions. After looking at my videos, after going through the videos, make some short notes or some short points, and then just go in-depth study, do the in-depth study. So you have ample of time in your hand, so you can do as much study, you can allot as much time as you can. So make sure that you don't take it lightly. Start your preparation really very well. Make a strong foundation of your preparation so that you can easily ace this exam. And trust me, this exam is not very tough. Just stick to my videos and practice the topics. Read the topics that are being given in your syllabus. Trust me, you will ace this exam with really very good marks. Okay, so let's go to the next question. Which of the following processes are involved in the horizontal gene transfer? So you have the horizontal gene transfer. I hope you have studied about the horizontal gene transfer in the bacteria. So here we have conjugation, transduction, binary fission, transformation, translational synthesis or transversion. So horizontal gene transfer includes conjugation, transduction and transformation. So I hope you know about all these three processes. If you don't know, I'll tell you. So in conjugation, basically the gene is transferred from one bacteria to the other with the help of a connecting link. Okay, so this is basically, it can be through a pili. Okay, so the genetic material is being transferred from one bacteria to the other. Okay, so this is the process of conjugation. In the process of transduction, the naked DNA is being transferred, and in the process of transformation, bacteriophage comes into role. Bacteriophage or bacteriophage, whatever you can call it. So bacteriophage or bacteriophage is responsible for the transfer of genetic material from one bacteria to the other. Okay, so this is the answer D, which is having all these options, which is conjugation, transduction and the transformation. Now the next question is match the following again. So we have some diseases and we have to tell the causative agents. So here I want to tell you one thing, that go through as much diseases and just cram the causative agents, the features that occur with these diseases, and just stick to some of the diseases which are being asked repetitively. So in this GATE exam, be sure to have questions from coronavirus. So I hope you study well about the coronavirus. You have ample of videos for coronavirus. This whole year has been consumed by the coronavirus. So make sure you study well about the COVID-19 virus, what it is, what is its morphology, where did it originate, what is its genetic material like. Okay, if I get time, I'll surely make a video on it. But if I didn't get a time, I'll ask you guys to please go through some videos. You can find so many videos on YouTube itself. Okay, or you can just Google search and read about it. You have so many articles on COVID-19. So just go through it. So here we will discuss about the diseases and the causative agents. So we have rheumatic heart disease, which is being caused by the group A streptococcus. Okay, then we have syphilis. Syphilis is being caused by Treponema pallidum, which is the option S. Then the genital wart is being caused by human papilloma virus, and oral thrush is being caused by Candida albicans. So the option B is the correct match to the following answer. Now, which of the following immune system components can function as an opsonin? So first we will talk about what is an opsonin. Opsonin is a substance which binds to foreign microorganisms or cells, making them susceptible to phagocytosis. So it's basically a substance which binds to the foreign material that enters into our body, and it makes that substance more susceptible, or more means it can be easily observed by the antibodies and can be phagocytosed or can be killed. So these are the opsonins. So I hope I mentioned the antibodies. So the components that can function as opsonins are the antibodies. Okay, they will stick to the foreign microorganisms or the foreign cells, and it will make them susceptible to phagocytosis, and hence they will be removed out of the body. Now, the oral polio vaccine, which is OPV, consists of live attenuated virus, killed virus, viral toxin, or viral capsid subunit. So I hope you know about this, what the vaccine basically consists of. So it basically consists of live attenuated viruses, which is a disease carrying virus which is being weakened in the laboratory so that it won't cause a disease, but it will cause a heightened or a strong immune response. That is why it is being used. So oral polio vaccine consists of live attenuated viruses. So that's all for this video. I hope you like this video. Please do subscribe to my channel if you haven't yet, and share my video with your friends who are preparing for this exam, who are your classmates. And thank you very much for watching. Stay tuned to my channel.